Hello, welcome to the Animal-Free Safety Assessment collaboration webinar dedicated to the replacement of the monkey neurovirulence test. We are starting in a minute. Hello again, my name is Laura Viviani. I am a consultant at Sciatica, collaborating with the Animal-Free Safety Assessment Collaboration and Humane World for Animals. And I'm here today with my colleague Jen Turner from Humane World for Animals. Welcome to this webinar that is the last of a long series of webinars that AFSA has set up in the last 3 years. This is dedicated to the replacement of the monkey neurovirulence test. Before starting, we would like to give you some information about this webinar.
Okay, yes, thank you all for joining us. I just wanted to go through some sort of housekeeping items. So in terms of the webinar today, all participants will be muted and their cameras will be turned off automatically. If you do need to ask us anything or comment, please do use the chat function in Zoom. So that's just down the bottom of the panel, the speech bubbles. And if you do want to ask a question, please use the Q&A function in Zoom for any questions. What we do have running is this Slido poll, and it would be really interesting to get people's feedback on the topics, the questions within this Slido. There's around 20 questions, but it would be really helpful for us to see your responses to that if possible. Okay, and I'll hand over to Laura again.
Thank you, Jen. Very quickly, if you are familiar with the work that Humane World for Animals and the Animal-Free Safety Assessment Collaboration has been doing since the last few years, it's a project that is supported by the Gates Foundation to accelerate the non-animal batch release testing for human vaccines. The project allows stakeholders to have dedicated discussion with experts in the field, joining local or a global workshop, or participate to initiatives like this webinar to learn a little bit more about the opportunities that exist or the ones that are in development, in order to make sure that you can plan accordingly within your own organization how to implement this type of opportunities. This webinar has been organized with the support of this project International Steering Committee, and some of the people that you are going to listen today are a part of that, and the others that are behind the scenes are have all our appreciation for all the work that they have been doing to support this webinar and all the activities that this project is moving ahead. For today, as Jen explained, we have a Slido that for us it's very important because we would like really to understand exactly what is your experience with regards to the monkey neurovirulence test, what are the struggles, the challenges, and it will help us also to understand what kind of additional activities AFSA and Humane World for Animals can set up in the next years to support your implementation of those the available opportunities. You will hear the speakers today sharing some concrete case studies about the use of monkey neurovirulence test and the opportunities to replace it with non-animal methodologies or with still animal-based methodologies, but using a different species. And you will also hear about the regulatory perspective, which is very important because things are being transforming also from that perspective. And it will be very important for you to learn how the regulations are going to be updated with regards to the use of non-animal testing for this specific test. as Jens mentioned, you are welcome to share your thoughts through the Q&A or the chat button that we are going to monitor through the whole webinar. Thank you for really being with us. And again, if you can really take some minutes to answer our Slido, it will be very appreciated. Without further ado, I would actually be glad to introduce the first speaker of today, that is Dr. Kostya Chumakov, that is going to give an important historical background about the monkey neurovirulence test and a little bit of the way forward. So Dr. Chumakov, feel free to come up online and share your presentation.
All right. Thank you very much, Laura.
Thanks to you.
Let me share the screen. All right. I hope you can see it now.
Yes, we can. Thank you.
All right. So my presentation will include several points. So first, I will talk about the objectives of this activity. Why do we need neurovirulence testing for live viral vaccines? And since the most information that is available up to date is based on the oral polio vaccine, I will talk about its history, and I will describe the monkey neurovirulence test that is being used. First, I will talk about the old CFR test, which was in the US Code of Federal Regulations, that is later was replaced by the so-called WHO test, the optimized procedure. Then I will talk a little about transgenic mouse test that was developed as a way of replacing primates-based neurovirulence by the transgenic mouse assay. And then I will talk about molecular methods. The first was MAPRIC, mutant analysis by PCR introduction enzyme cleavage, that was developed in our laboratory more than 30 years ago. Then I will talk about the next generation sequencing, or deep sequencing, high-throughput sequencing, massively parallel sequencing. There are a lot of names for this new technology. And finally, I will talk about the use of whole genome single nucleotide polymorphism profiles as a way to ensure consistency, molecular genetic consistency of live viral vaccines. So why do we need testing to test vaccines for neurovirulence? So live vaccines, of course, are they inevitably are prone to mutations that can lead to some adverse consequences. Live vaccines were created by selection of attenuated versions of the pathogen. And of course, if you can select for attenuation, then it can select for de-attenuation. So, and since attenuation generally reduces virus fitness, so growth of virus actually favors selection of the non-attenuated versions. And that's why you need to grow virus in a very strictly defined optimal conditions. And if you don't really strictly adhere to the conditions of virus growth, then it can lead to increased virulence. And it's not only virulence, essentially, you can also expect that mutations can take place in the protective epitopes, reducing viral efficacy. So in general, as we all know, manufacturing consistency is a cornerstone of good manufacturing practice. And certainly it is important to maintain stability of critical quality attributes. And of course, neurovirulence is one of those key attributes. So I will talk about polio vaccine for a bit. So it was developed by Dr. Albert Sabin in the mid-1950s. It's made of weakened attenuated virus selected from pre-existing variants of the virus from populations in the wild type virus stocks. What is good about this vaccine, that it is administered through the natural route of virus transmission, meaning orally, and it induces comprehensive immunity, not only humoral immunity that consists of circulating antibodies, but also mucosal immunity. And it also creates a herd effect through transmission to contacts of the primary vaccine recipient. And of course, this vaccine was instrumental in the almost complete elimination of the virus worldwide. So all countries except for the 3 Scandinavian countries used it, and some are still use it. So here is the reprint from the paper published by Dr. Sabin in 1956, in which he demonstrated that there is a whole range of virus particles present in the population, ranging from essentially it's like a 6 or 7 more, actually 13, I think, orders of magnitude of neurovirulence. So this is a dose that results in paralysis in monkeys. So you see that the most in the brainstem, it takes much more virus than in the lumbar cord neurons to paralyze a monkey. And this is a cynomolgus monkeys. And in chimps, actually, it's very different, because viruses that are highly pathogenic, actually paralytogenic in cynomolgus, are not paralytogenic in chimps at all. So that tells us that this test actually is not strictly a test for safety of the vaccine, but rather it's a consistency test. Because those, so cynomolgus monkeys and test in these monkeys is exaggerated. So these viruses that are paralytogenic in monkeys, they are not paralytogenic in apes and are not paralytogenic in humans. So this is the way Dr. Sabin derived his virus. He started with the wild virus, he made passages in different cell cultures, then he plaque purified them, then he tested again in monkeys, and then he created a stock that was distributed to Merck, and that's why it's called Sabin Original Merck, and this SO+1, Sabin Original +1 passage. And this stock actually is the source of all type 1 vaccines that are being used right now. Dr. Sabin, early on in his work, he actually saw that passage in cell culture and in the gut of vaccine recipients leads to loss of attenuation. That's why he required all manufacturers to test every single batch of vaccine for neurovirulence in monkeys. And that it was done since then. It was his absolute requirement for vaccine manufacture. So first, this Code of Federal Regulation actually prescribed this type of test, which is based on the protocol that Dr. Sabin developed. So monkeys were, of course, screened for the absence of antibodies to polio. They were randomized between the test and reference group and inoculated directly into the anterior horns of the spinal cord with 2 doses of vaccine. Then the monkeys were observed for 17 to 22 days, and most of them, they never developed paralysis. So actually, they all felt pretty well. But at the end of this observation period, they were sacrificed, and histological examination was performed of the spinal cord and brainstem. Lesions were compared between the test and the reference group, and if the test group showed a higher lesion score than in the reference vaccine group, then the lot was actually considered unacceptable and was not released. And what's also important that this test was performed by both vaccine manufacturer and national regulatory laboratories. In the mid-1980s, World Health Organization organized an international collaborative study to actually look at this test, and this collaboration resulted in an optimized procedure that is now being called WHO monkey neurovirulence test. So it's simplified to some degree, because 2 groups of monkeys, of course, were inoculated similar to the CFR test, and for type 3, 24 monkeys were used for vaccine lot and 24 for the reference. For type 1 and 2, only 12 monkeys were sufficient. So they were observed for 17 days, and then histological lesions were examined, and they were scored by a standardized procedure, standardized method, scored on the scale of 1 to 4. And the same thing actually as in the previous test, a lot passed if the lesions are not greater than in the reference vaccine. So overall, there were 200 monkeys used to quality control just one lot of trivalent vaccine. And of course, this was it was not a good test, not only because it was not very sensitive, and the results were actually, like most animal tests, actually were very variable, but it was definitely extremely expensive and inhumane. And again, I want to stress that this test is not a product, it's not a safety test, it's a product consistency, because there is no evidence that failure in MNVT leads to unsafe vaccine. The only thing it indicates is the breach of manufacturing consistency. If the MNVT is increased, you may want to look into why it happened. It means that there may have been some breach in the production conditions, consistency, and in any case, we don't want a virus to drift into the higher neurotropism. Therefore, there was a lot of interest in trying to develop some alternatives. And one of these alternatives was developed, again, it was developed in our lab. It was transgenic mouse test that actually is still an animal test, but at least it was not using, more psychologically sensitive primates. And at this point, I want to pay homage to Dr. Nessa Levenbook. I think she single-handedly started this ball rolling. In her lab, monkey transgenic mouse test was developed by Dr. Eugenie Dragunsky, and actually she invited me to join the Food and Drug Administration in the late 1980s to start working on the molecular alternatives. Here is their seminal paper on transgenic mouse test. But what actually caught her attention was that in 1985, Dave Evans, in collaboration with Phil Miner and Jeff Almond and Jack Mizell at NIH, they have identified a molecular mutation in the genome of type 3 poliovirus that led to increased neurovirulence. So this was the main culprit of this reversion to virulence that took place during inappropriate virus condition growth and in the gut of vaccine recipients. And here you see that even just after a few days after inoculation of vaccine, actually immunization, in this case it was a son of Phil Miner, 3 days after he got the vaccine, he started shedding mutated virus. So this caught Inez's attention, and she was a pathologist, she was not a molecular biologist, and so she invited me to join her group and to start working on the molecular methods. So we very quickly developed this procedure. Here is the mutations. This is the domain 5 of the 5' untranslated region of polioviruses. Interestingly, in all 3 serotypes, mutations that led to increased virulence were located in this domain 5. So we developed a PCR-based technique. Actually, we amplified a short stretch of viral genome and digested with the restriction enzyme. So by looking at this intensity of this spot digested by a restriction endonuclease that cut this DNA at this point at the site of mutation, we could quantify the amount of mutated virus in vaccine lots. And to our surprise, actually, we realized that every single batch of vaccine contained this mutation. But the difference was that the acceptable vaccine lots that passed monkey neurovirulence test, they were located in this low range of the content, so essentially below 1%. And all lots that we were available to us, that failed monkey tests, they were in the higher range, and there was almost a linear correlation between the content of mutants and histological lesion score. And the reference vaccine that was used for monkey neurovirulence test is shown in yellow. It was right in the middle. So it was astonishing. I mean, we were just amazed that there was such a good correlation that, of course, opened the prospects of using this technique to predict the outcome of the monkey neurovirulence test and eventually replace it. So, of course, we went to Geneva, to WHO in 1991, and here you see Inessa and I sitting on the shores of the Geneva Lake after this meeting that resulted in starting of a collaborative study that was successful. It took several phases of a collaborative study, but at the end of the day, WHO committee on the expert committee on biological standardization approved MAPRIC as an in vitro test of preference for lot release. Sadly, to our disappointment, the ECBS did not agree that MAPRIC can replace any monkey neurovirulence test because of the concern that first, it was something very, very new. It was the committee was completely unfamiliar with these molecular techniques. And second objection was that, of course, we look at only one mutation. Even though it's an important mutation, this mutation is critical for a reversion to virulence, but we could not guarantee that there is no other mutation in other parts of the genome that could also result in higher virulence. So, and it was a step forward, of course. RCT40 marker was actually removed from the requirements, and vaccine lots that failed monkey MAPRIC should not be tested in monkeys because it was at least some saving on the life of monkeys. So I already told that what were the reasons for ECBS not agreeing with the replacement of the monkey test, but there are also some other reasons. It's very technically challenging, I would say. It requires very highly skilled personnel, standardization of all the laboratory procedures. It requires radioactive isotopes, which nobody likes. And in some labs over the time, there's still unexplained baseline drift, meaning that the reagents that were used and developed standard reagents perhaps are not as stable as we initially hoped. So that's why there was a push for trying alternatives to MAPRIC that could actually do the same thing but better. And so we started looking at different alternatives, and then I think this paper was published in 2010 about the use of massively parallel sequencing. So basically you sequence multiple individual molecules of nucleic acids, and we used a very old 454 technology. At the time, the coverage per each nucleotide was roughly 10,000. At the time we thought it was great. Now we can do it much better. But most importantly, we realized that comparing single nucleotide polymorphism profiles, essentially it's here is the entire genome, and these bars, they show the content of different polymorphisms or mutations, if you wish. And this is a failed vaccine lot, and this is a passed vaccine lot. You can see that there is a big difference. And if you look closer at the results in this critical 5' UTR region, you see that the reference materials for MAPRIC, this is the passed reference, and here is the content of 472C, and this is a failed. And this is a US neurovirulence reference. So there is a very good agreement between the results of MAPRIC, quantification by MAPRIC and by the NGS. And we approached the WHO, and the WHO recommended a collaborative study, and it was led jointly by CBER FDA and NIBSC, which is now called MHRA. And I'm not going to talk much about that because I don't want to steal Javier Martin's thunder. He will be talking about it at this webinar this morning. So essentially, just a summary, this approach was approved by CBS, and Javier will tell you how it was done and show the results. But the comparison of vaccines made by different manufacturers actually showed that there is much more in these profiles than just 472. Because, I mean, here are 4 different manufacturers, and you see that all the profiles are different. So how do we compare the profiles? So if you have consensus sequences, you just align them and then you count positions at which they differ. So this is a measure of genetic distance between 2 consensus sequences. But what if you have not consensus, but actually the content of all mutants in all positions? So theoretically, at each of the genomic position, you can have 3 SNPs, because there are 4 nucleotides, so there could be 3 mutations. How do you condense all this rich information into one metric that can be compared, and you can judge whether this difference is significant or not significant? So there are some ways to figure it out. I'm not going to talk, I mean, there is no time for me to describe all the math behind it, but at the end of the day, it's very easy to compute this distance between 2 profiles. And here you see several dozens of vaccine lots, their phylogenetic relationships. So it's like a genealogical tree. So these are seed viruses, 2 different seed viruses, or 3 actually seed viruses. So these are manufacturer B, and the vaccines made by manufacturer B, here it is. So these are production lots. They are all clustered on the same branch of the tree. So this manufacturer A produces vaccines that are not really clustered pretty well because they were made from different seed viruses. And by the way, the same is true for manufacturer B. Here is the one group and here is another group. So essentially, by comparing the profiles, SNP profiles, you can tell not only which manufacturers made the vaccine, but also tell that vaccine is made from a different seed virus. Before I finish, I will try to show you how this can be done for lot release and for quality control of vaccine lots. So here is the historical data. So what we propose actually is that when a manufacturer is establishing consistency of their manufacture, and this is done anyway, I mean, even before you can license the product, you have to establish consistency. You produce several lots, you test them in whatever methods you use, it's MNVT or something else. So on this establishment phase, you test vaccines by both monkey test and by NGS. So here are 5 lots. Let me get rid of this on my screen. Okay, so there are 5 lots. Each lot was tested 5 times. And here is the content of mutants, positions in which mutants or SNPs, nucleotide polymorphism, exceeded 1%, because anything below 1% should not be considered because it's a noise. And here is the lot under consideration. Let's call it X. As you can see, there is a content that differs in these lots, and here I highlighted in yellow the position at which the content of mutants is statistically significantly different in the historical background and the vaccine lot under consideration. All other mutations are not statistically significant. So these differences are very minuscule. So, yes. So this is the same thing. I just to zoom in into this part of the table. You see this 2493. We know about this mutation. It appears in many different vaccines. In some vaccines made by some manufacturers, this mutation is 100%, and still there's nothing wrong with it. It's the vaccines with 100% 2493 were used successfully without any adverse effects, reactions. So this is lot X. So let's look at, and we conclude that this is acceptable. There's nothing suspicious in the profile. Lot Y, it's more complicated. Again, we see the same thing to 493, but in this case, in the lot under consideration, the content is lower. So obviously this is acceptable. And in this case, 3262 mutation, it's a silent mutation. It's statistically significant difference, but the difference is, I mean, it's just because it's very tight. It just speaks to the precision with which we can determine the content of mutants. So, and I'm telling you that this mutation is not important, and I will in a second tell you why. Again, we conclude that this lot is acceptable. Now, a more complicated case. There are several mutations that are statistically significant here in the 2C region. But again, the content is lower, so we're not concerned about this much. And this mutation in 2A protein, which is missense mutation, and again, it's pretty tight, but still it's significant. So, and it's important for us to understand that statistical significance is not the same as biological significance. So how do we address this? If we identify the mutation that is different, so can we judge on whether it's important in terms of virulence and other properties or not? So what we suggest is that here is the summary of all mutations that were present over the many years by many manufacturers, were present in vaccines that were judged acceptable by monkey neurovirulence test. And you can see in some cases the content is close to 100%, 98. Well, I mean, it's never 100 just because of the accuracy of determination. And here is the list of mutations that it's like a white list of mutations. If we find in a vaccine a mutation that actually matches this list, then we should not be concerned. So that's why the algorithm for lot release by using SNP profiling could be like I just described. So we compare the content of mutants in the entire genome. We look at the potential biological significance, some adverse information that we can deduct from this profile. And if we judge that the lot is acceptable, then we release it and update the database to include the information about this new lot. So this is pretty straightforward, and I think that at least we all hope that this will be accepted by the CBS. There was a collaborative study, and again, Javier will talk about it. So we believe that this could be the first case of NGS, whole genome SNP profiling, being used as a consistency test that essentially can replace monkey test, because monkey test is much less informative and it cannot detect some mutations that are unrelated to virulence. It just looks at the one phenotypic property, while this one looks at the entire wealth of genetic information that's contained in the viral genome. So I think this is my last slide, and I'm ready to answer any questions that you may have.
Thank you very much, Dr. Chumakov. At the moment, we don't have questions into the Q&A box. but I would welcome all our attendees that had any questions there. We can review them during the panel discussions, or if there are some questions that can be answered in written into the Q&A, we will do so. So for now, Dr. Chumakov, thank you very much for your introduction and introducing some of the aspects about the availability of alternative methodologies for the polio virus vaccines. And I would like right now to introduce our next speaker. It's Dr. Karine Lozhanov from Sanofi. She is going to give you some concrete, a concrete case study for the yellow fever vaccine. So Karine, the floor is yours. Thank you for being with us.
Thank you very much, Roa. Can you see my screen?
Yes, yes, not yet on the presentation mode. Yes, now it's there. Perfect. Thank you so much.
So thanks for giving me the opportunity to present you the challenge with the neurovirulence test for yellow fever vaccine. So first, a little bit of background regarding yellow fever disease. So this is a viral disease that is transmitted to humans by the bite of infected mosquitoes. It is prone to epidemics and it's preventable with a vaccine. Currently, there are 34 countries in Africa and 13 in Central and South America that are endemic or which have a region that are endemic for yellow fever. The vector for yellow fever are mosquitoes, Haemococcus or Aedes species. They are transmitted after a blood meal from an infected animal. And after 10 days incubation, yellow fever is secreted in the saliva of the mosquito and is transmissible to another host. There are three types of transmission cycle. The sylvatic or jungle cycle, it's when yellow fever is spread through monkeys bitten by mosquitoes and wild mosquitoes that pass over from monkey. Then there is the intermediate cycle between mosquito, human, and primate, and there's the urban yellow fever cycle that can cause large epidemic when infected people are introduced within heavily populated areas with high mosquito density and where people have little or no immunity. So the causative agent of the disease is the yellow fever virus. This is a prototype of flavivirus. As just mentioned, it's an arbovirus. There is only one serotype of yellow fever. There is 7 genotypes, 5 in Africa and 2 in Latin America. It's a small envelope virus. Its genome is made of a single-strand RNA, positive strand, which encodes for a polyprotein that is later on processed in 10 proteins, whether they are structural or non-structural proteins. Regarding the symptoms of the disease, most of the people infected with yellow fever likely do not seek medical attention because they have minimal or no symptoms. However, for the people who will develop symptomatic illness, the incubation period is typically from 3 to 6 days following the bite from the mosquito. The initial illness is not specific. It's fever, chills, headache, myalgia, prostration, nausea, vomiting. And after a brief remission of up to 24 hours, approximately 12% of those within the acute phase will enter into the toxic phase with a more serious form of disease, which is characterized by jaundice, hemorrhagic symptoms, and eventually shock, multisystemic organ failure, and the case fatality rate for those who enter in the toxic phase is quite high, between 20 to 60%. So there's no specific treatment for yellow fever, so it's only supportive medical care that can be provided to symptomatic patients. So the best thing to do is prevention, the prevention through the vector control and through the vaccination. So thanks to a 1-year century work. There are vaccines that are available, extremely efficient vaccine. 1 single dose of vaccine is sufficient to provide what is now considered as a lifelong protection, and there is no booster dose that is required. Currently, there is 6 vaccine manufacturers worldwide, and all the currently approved yellow fever vaccine are live attenuated viral vaccines that are derived from the 17D strain and the 2 substrains 17D and 17DD and 17D204. All those vaccines are produced on embryonated chicken eggs, and there are currently 4 WHO pre-qualified vaccines. There are some development, and within Sanofi we are developing a new alternative vaccine produced on Vero cells. So as mentioned, the development of the vaccine started a century ago, in parallel with the work of Max Taylor on one side and the work of Institut Pasteur on the other side. So there was 2 different developments that were established, one for the AZB strain and one for what was named the French neurotropic virus. In the 40s, there was mass vaccination with those 2 types, the French neurotropic virus being massively used within a French-speaking area of Africa. In the 50s, 60s appears the post-vaccination concern. So there was a little bit of concern with the 17D for children below the age of 9 months with some neurological adverse events that were really sporadic, but they were more concerned with the French neurotropic virus with a higher rate of post-vaccination encephalitis in children. So in a first step, there was the vaccine was not for children below the age of 14, and then it was discontinued in 92. So currently, all the vaccine in use are derived from the 17D strain, and it's the strain that has been recommended by WHO, PAO, and UNICEF for the routine vaccination program. So this is a view of the isolation of the 2 vaccine strains. So the French neurotropic virus was derived from a French viscerotropic virus that was passaged on mouse brain. So this French neurotropic virus lost viscerotropism, meaning the infection of the liver and the mosquito competence. And the vaccine that was used was passaged on mouse brain. So, as already mentioned, with the increased neurovirulence rate with the encephalitis of children, it was contraindicated for below the age of 14, and it was discontinued in 92. Regarding the AZB strain and the 17D strain, so this is the work from Max Taylor and co. So the isolation was from a Ghanaian patient with a mild yellow fever. It was passage in monkey for 53 passage, and then there was many other attenuation passage on different type of substrate, mince mouse embryo, mince whole chick embryo, mince chick embryo, and without brain and spinal cord. Throughout those attenuation passage, the attenuation was followed both through the passage in mouse with intracranial inoculation, and as well as in monkey. The 17D strain loss is viscerotropism and neurotropism in monkey. And because of this history, now people have to test yellow fever vaccine for both neurotropism, for viscerotropism and immunogenicity, and this is done through the test that is named test in non-human primate. This test is described in detail both in WHO technical report series document, as well as in the European Pharmacopée dedicated to yellow fever vaccine. This test is composed of 3 parts, as mentioned. viscerotropism, which correspond to viremia, immunogenicity, and neurotropism. For each of the test articles to inoculate, you will need 10 monkey, and you will need also 10 monkey at minimum for the reference vaccine. It's through intracranial inoculation of a defined dose of virus, which correspond to 5 to 50 vaccine doses. Following this inoculation, the viremia will be checked on day 2, day 4, and day 6 post-inoculation. And this is to ensure that there is a limiting amount of circulating virus following the inoculation. 30 days after the inoculation, the sera will be tested for immune response against yellow fever. This is also a way to ensure that the inoculation were also properly done and that the monkey were indeed immunogenized this way. During the 30-day of post-inoculation, the monkey are observed for clinical sign on a daily basis. And there is a scoring attributed to each clinical sign. And on day 30, the monkey are euthanized, and it's an histological evaluation that is performed. It's through the examination of the cervical and lumbar enlargement of the spinal cord and the specific structure in 5 levels of the brain that overall the neurotropism of the seed lot to be tested is assessed. So this is how the test is to be done these days, but it started a century ago with the work of Max Taylor. We identified that monkey were a way to assess the virulence of yellow fever vaccine. In 42 case encephalitis in Brazil were studied in monkey using intracerebral inoculation. And this work ended with the requirement mentioned in 42 to test yellow fever vaccine in monkey, and this was made as a test requirement in 45. Dr. Schumacher introduced Inessa Leeuwenhoek earlier, and she made also a really great evolution within this test. So initially, there was only the viremia, the immunogenicity, and the daily clinical observation that were performed without any scoring. And this is really not something that is precise, and it's a pity to use monkey to have such a variability. Throughout the study that she conducted within the 80s, she was able to make a really nice evolution of the monkey test and improve this test by introducing a scoring system for the clinical sign, but more importantly, by defining the histological evaluation one should conduct. So with this associated scoring scale, similarly, the 1 to 4 scale, there is a quantitation of the clinical observation, which really improved the WHO procedure. The test variability was greater on the clinical observation scoring than on the histological observation, but at least there was a frame. And indeed, we highlighted that the clinical scoring is something that is extremely variable, and especially in non-human primate, and that although the histological scoring are really consistent, the clinical score can really vary, and we highlighted that while we were using the same reference material between a study that we conducted in 2011 and a study in 2018, where we saw really some statistical difference, between the two studies for the same material. So that is to say that with this variability of the test in non-human primate, we risk to have some seed material that are good to make viral vaccine that will be disqualified because they failed the test for improper variability of the clinical score. So currently there is there was an amendment of the testing. The test is still performed the same, but at least it is currently acknowledged that the clinical evaluation may be imprecise. So is there any alternative to this non-human primate test for yellow fever vaccine? And again, this yellow fever, this non-human primate test was defined specifically for yellow fever. So there was some assessment with the hamster. The hamster model can show viscerotropic disease. So that was a first good approach. However, those hamsters are not able to identify some viscerotropic disease from viruses that were taken from yellow fever vaccinated people who presented viscerotropic disease. So not such a good model. Then there was another approach, which is still with a smaller animal, the mouse, either using mouse that are deficient for the interferon cascade, so deficient in the interferon alpha and beta, the A1 to 9 mice, through subcutaneous infection, or the approach that we follow within Sanofi, which is the intracerebral infection of OF1 mice. Indeed, with this intracranial inoculation, depending on the level of neurovirulence, we may see different symptoms, clinical sign, or death. Traditionally, yellow fever, and including the vaccine, 17D, will kill adult mice within the 3 weeks following the inoculation. But you can score the clinical sign and see if you are losing some neurovirulence depending on the strain that you made, and this I will describe this in the next slide. But we also have a different family of flavivirus within Sanofi, and we could see that we could use mice of different age to identify neurovirulence of different grade, meaning that although yellow fever is known to be highly neurovirulent, and for this we use adult mice, 4 weeks old. For flavivirus construction, which are of lower neurovirulence, we have to go to younger animal, suckling mice, or even infant mice, infant or suckling mice, to really identify some fine neurovirulence. So regarding this adult mice test, I mentioned that we are developing internally a new alternative yellow fever vaccine that is produced on Vero cells, and we assess its neurovirulence in the adult mice. This vaccine was derived from the YF-Vax vaccine, and we have a scoring system. Basically, black, the mice is dead, red, it's some severe observation clinical symptom, and the green mouse is a happy mouse that is alive. And if we have some mortality with the yellow fever vax material, with the material that we develop on yellow, the mortality is highly reduced, and we have a safer profile observed on this small animal. So potentially the mice can be an alternative to the non-human primate test. It will be even better if we can move to a fully in vitro approach, whether it is in cellulo. So there was some pilot study with blood-brain barrier mini brain that were set up, but this is not yet established, and there is also the possibility to directly look for the sequence of the vaccine. So from the work of Charlie Rice in the 80s with the first sequence of yellow fever to the more recent use of high-throughput sequencing, the attenuation-related mutations have been identified and are shared all along the genome. Through the comparison between the AZB and the 70D204 genome, there is 67 nucleotides that are different, and it's encoding something like 31 amino acid substitution. However, to date, the specific amino acid contribution to the attenuation of the 17D have not been elucidated. So the role of each individual mutation in the attenuation is not known. However, and this is from the recent use of high-throughput sequencing, it is highly considered that the relative quasi-species structure is a plausible correlate of the attenuation of live viral vaccine, meaning that the vaccine strains are far less variable and present far less quasi-species than the wild-type strain. So regarding the alternative to non-human primate tests, so in the absence of clear understanding of the 17D attenuation, the relevance of in cellular approach as an alternative to the non-human primate test might be challenged. However, considering the sequence data alone might also appear difficult, since the importance of each attenuation-related mutation within the overall attenuation is unknown, and the interaction between mutation within the overall attenuation is also unknown. In-depth sequencing data should be considered within an overall characterization package that is both assessing the genetic and phenotypic characteristic of the viral population. So our proposal is to demonstrate the consistency of the viral population, both genetically and phenotypically, with previously qualified material, such as a reference slot, and this better than just running systematic non-human primate test. I want to highlight that within the recently provided for comment draft of the guideline for the replacement or removal of animal tests for the quality control of biological product by WHO, it was highlighted that the precise mechanism of genetic basis of neurovirulence of yellow fever vaccine are poorly understood, but that the level of residual neurovirulence of an attenuated virus is still determined by the viral genome sequence. So as a result, validated molecular methods are suitable alternatives to the in vivo neurovirulence tests for the control of yellow fever virus seedlot. So it's by leveraging previous non-human primate test results and the consistency of the sequence that potentially we can avoid performing any more non-human primate tests. So to conclude, yellow fever vaccine are a very successful live attenuated vaccine. With a single dose, you can have a high and early immune response that is long-lasting and with a good safety profile. However, the test in non-human primate is still required for seedlots. And while the relevance of alternative in vitro model might be difficult to demonstrate, it's the consistency of the genetic and phenotypic characteristic of the viral vaccine with previously established material that should be considered as an alternative to neurovirulence test. And with this, I thank you for your attention.
Thank you very much, Karine, for your presentation. At the moment, we don't have the questions in the Q&A. I really encourage the attendees to add their questions. In the meantime, I'm happy to move to the next speaker of this webinar, which is Dr. Javier Martin from MHRA, that is actually going to nicely continue what Karine has started to introduce, like the next generation sequencing. So Javier, the floor is yours, and thank you for being with us.
Okay. Hello, everybody. Can you see my screen? Yeah.
Yes, we can.
Okay.
Javier. Thank you.
Okay, I'm going to expand on the previous two presentations, particularly the one from Kostya Chumakov, who nicely introduced the subject, because my talk will be mostly dedicated to poliovirus vaccines. And as he mentioned, Sabin and Berger prepared the live attenuated OPV strains by successive passages in vivo and in vitro, and different step selections on viruses with low neurovirulence in monkeys. As a consequence, by using mostly wild poliovirus strains, mutations accumulated in the genome providing attenuation. So mutations, there is a lot of works through many years, including my lab, or the lab, my previous lab directed by Phil Minor, in which mutations responsible for attenuation were clearly identified for the C poliovirus serotypes, and particularly mutations in the 5' domain non-coding region were prevalent in the 3 types, and they were the strongest determinants of virus attenuation. This is shown in this slide, the map in the same domain, which is shows how remarkably similar the 3 strains were prepared. Unfortunately, with the years, it was soon found out, but that these vaccines were not stable. So, in fact, only a few days after administering a vaccine to a child, the virus reverts, particularly reverts in mutations in the 5' end, increasing virulence in monkeys, which is the test conducted for batch release. So, for that reason, it was very important to maintain a very good test and a test for every batch to make sure the virus had not reverted. So, as mentioned here, this is summarized here: OPV prepared by serial passage of wild poliovirus strains. The virus is weakened by incorporating mutations. There is a very low risk of disease caused by the vaccine. The potency assay is a titration assay in cell culture, which determines the number of infectious viruses present in a preparation, and the safety or consistency test, as Kostya mentioned, needs to measure reversion to virulence. Through the years, I mean, the monkey test was it is the gold standard test, because in fact it was the test that was used to develop the vaccine, and it continues to be the gold test, but a number of tests have been developed as alternative to this one, so it has replaced the use of monkeys, thankfully, for many of the steps during vaccine production. However, still mice expressing the human poliovirus receptor still used, and also for some steps we could replace, as Kostya mentioned, these neurovirulence assays for a molecular test measuring the mutation reversion at the 5' end site. And this is summarized here through the years. Different tests were required, different stages, and then now monkey test is only used when a new vaccine production starts production. So basically, it's not used any longer. Ideally, and this is the subject of this webinar, we could replace all these tests using animals by tests using molecular assays, next generation sequencing, or HTS. So through this talk, please consider these two terms identical, NGS or HTS. One of the points about also about the animal test is not only unethical and difficult to accept that, you know, injecting animals that develop disease and paralysis, but also they are very complex tests that require very careful procedures and very skillful. So it's not easy to develop. In fact, for example, a transgenic mouse test might take from 1 to 3 years to be implemented in an institution. The MAPREP test, Kostya mentioned, which is based on restriction enzyme cleavage. So these are enzymes that specifically cleave DNA at specific sequences. So by using an enzyme that cleaves the molecule containing the mutation, you can measure the amount of reversion of that PCR product. And obviously, this is also quite complex and delicate test because you need a lot of controls. PCRs can be contaminated. You are measuring a very low amount of reversion, so also a very complex test. So with the advent of a high throughput or next generation of deep sequencing, we hoped that by using this, you could characterize live attenuated virus, not only polio, but for others like yellow fever we have seen in the previous talk, and replace the test, the MAPREC and the neurovirulence test. And there is early evidence from papers from the Chumakov's group and from Sarsi from Sanofi, in which it was clear that potentially using NGS would be a very good test to be used to characterize the consistency of vaccine production. So this is just a short explanation of why this is the case and how HTS differs from traditional sequencing methods. RNA viruses actually exist as viral quasi-species. This means that they have a population structure in which they don't have a single molecule with a single sequence, but multiple sequences with a low number of mutations in each from each other, that overall they make a consensus sequence that's defined as the sequence of that virus. But there are groups of genomes, of thousands of very different genomes containing mutations. So, and this is the consensus sequence that is obtained by the traditional methods like Sanger. You only obtain a single sequence per sample that you sequence. And that is kind of the summary of all the sequences present in that preparation. But with a high throughput next generation sequencing, you could actually sequence each molecule in your viral population, and then you can count the proportion of nucleotide at each position. So that's what is called SMP, single nucleotide polymorphism. You can measure the SMP profile at each nucleotide position of the whole genome. And therefore, it provides you a much higher resolution for comparing viruses. This is one example. You can see on the right the whole genome profile, 7,500 nucleotides approximately, and the presence of SMPs, mutations at different proportions in the genome, and how this, for example, these two virus preparations differ of containing different SMPs. So by showing this potential of NGS as a potential for a test to replace or to characterize the consistency of vaccine production of live OPV, we worked together with FDA and many other groups, manufacturers, and PATH and WHO, to try to evaluate this with a possibility with replacing eventually all neurovirulence tests with just tests using molecular sequencing. So we had two phases. The first phase, we evaluated the use of NGS as a replacement for MAPREC. This is MAPREC, if you remember, measures the mutation, single mutations, which are the main determinants of attenuation. And so we just use the single mutation in the 3 serotypes to see if results were comparable by using NGS. We found this was the case. Very good correlation between MAPREC and NGS. You can see here in the 8 labs that participated in the type 3 study. So with this, it was concluded, and ECBS accepted that MAPREC could be used as a replacement for, NGS could be used as a replacement for MAPREC. Then we did studies for type 1 and 2, and the same result happened. So for the 3 serotypes, we established that NGS could be used as a replacement for MAPREC. As you can see, very good correlation, even at this low level of mutation proportions. So the second phase, we want to go further, and because obviously by only measuring one mutation, it would be not sufficient to replace an animal test, because you could have different mutations arising during production that could be responsible for changing in properties, not only in neurovirulence, but maybe antigenicity, immunogenicity. So for this, we ask ourselves, can whole genome sequence SMP profiles be used as a replacement for a neurovirulence test, eventually removing the need for animal testing? And this would actually be useful for both for OPV to be used as a live vaccine, but also for OPV seeds to be used for inactivation. So there are two vaccines, OPV and IPV. Inactivated vaccine uses wild strains or used to use wild strains traditionally, but they could be replaced with using live attenuated to add another safety step. So we then analyzed this, and a lot of work was conducted in both FDA and our lab at NIBSC. This shows the past history of OPV seeds, showing that for type 1 and 2, they were quite similar in the different labs, different seeds for different manufacturers, following similar processes between master working and lots. But for type 3, there were some changes in production in the 50s, 60s, because there were too many vaccines failing the monkey test. So it was decided by manufacturers to actually purify the original saving, original strain, and prepare new seeds that would hopefully have improved neurovirulence profiles, and that was the case. But the issue is that different manufacturers did it differently. So this ended up in vaccines being different between themselves in terms of sequence profiles. So we applied whole genome sequencing to study these mainly 3 different groups of vaccines, those based on the original saving, based on the another seed prepared by plaque purification or RNA purification, and another seed prepared is called by the Japanese seed prepared in Japan. So what we did was characterize whole genome profiles of the original vaccines, and then the original seeds and different vaccines produced with these different seeds. And we found a very good consistency within the seed used for vaccine production. You can see here in different colors that the different vaccines from different groups have common SNPs or variations that are consistent between the different products, even from different manufacturers. So the summary is here. Whole genome NGS analysis demonstrated high consistency in vaccine production of most OPV products by different manufacturers. Whole genome SNP profiles were highly consistent between vaccine products from the same manufacturer. Different vaccine seeds and associated products were found to contain unique SNP profiles, and I have to say all of these products had passed the neurovirulence test in monkeys and some in mice, so were safe to be using in humans. So those SNP profiles can be considered safe. Vaccine products for manufactured using the same vaccine seed were found to contain common SNP unique to that seed, but also manufacturer-specific SNPs were identified, often associated with the cell substrate used for vaccine production. For example, vaccines made with the same seed using monkey kidney cells, vitro cells, or human dipple cells had specific mutations associated with each of these cell substrates. These results strongly suggested that whole genome NGS analysis had a great protection as a QC test for OPV, measuring vaccine production consistency and potentially replacing the neurovirulence testing using in animals. So, and this was found both in my lab and Kostya's lab using many products from several different manufacturers. And also, this illustrates an example of how this analysis could identify vaccines that are not good, not safe to be used in humans. This is an example of vaccines produced by 3 manufacturers, 1 and 2, produce safe vaccines, and you can see that the production is consistent in terms of the proportion of mutations at some particular positions from master seed to vaccine lots, where the manufacturer 3, there was a massive increase of some mutations, and even new mutations appear that were not identified in the seed through production working seed and the bulk. So you can see here clearly that this profile was very different to what is expected, and in fact that they were shown to be neurovirulent using the animal tests. So the aim of we with all of this in mind and all this background experience, we are conducting a collaborative study to support this phase 2, and the primary aim of the study is to establish reference reagents to be used in NGS methods to monitor the consistency of production of OPV and the characterization of virus bulks used for the manufacture as IPV prior to virus inactivation. The objective was to establish a reference reagent suitable for measuring MAPREC-specific single mutations and/or whole genome sequence analysis. The study is also focusing on providing appropriate test formats and bioinformatics analytical processes for establishing assay validity criteria, so pass-fail. And overall, the study will provide further scientific assessment of NGS as a replacement test of animal neurovirulence tests for vaccine lot release. So this test is ongoing. I don't have time to go through the preliminary data. We are preparing a report to submit to CBS, but we are finding very good and very promising results. For example, this shows the concordance of results between 2 different labs measuring mutations of the candidates for this collaborative study, candidate virus preparations that contain mutations at different levels. And you can see how well results from 2 different labs correlate. So basically the study, the main aim is to develop or establish standards that labs can use. For example, a standard to measure the background mutation. So it's the virus that expected to contain mutations at very low proportions, and then all other viruses that contain mutations at different levels, so we can ensure that labs can measure mutation profiles containing mutations at different proportions. As you can see, for example, in this slide, that some of these references contain mutations at nearly 80%, 100%, 30%, all wide range of mutation profiles. And I want just to finish with a couple of slides. I mean, it has been mentioned here that this is also potentially useful for yellow fever, and this is yellow fever passage history from a CB, is the original virus from which 70 was developed. So we could characterize many of these strains and try to understand the mutation profiles and how, again, molecular consistency could be used for establishing pass-fail and to be used as a QC test for yellow fever. This, as mentioned as well, it might require longer time because we don't have such good information on the role of the different mutations in the attenuation of the vaccine, although for consistency, that might not be totally relevant. So as long as maybe consistent profiles are obtained from different vaccines, that might be sufficient. And so we developed, mainly my colleague Mansi Majumdar in my laboratory has evaluated the possibility of using this NGS for this purpose, and it has very good potential. You can see we identify different SNPs. I mean, you can see here, in this case, the genome is longer, so technically the analysis is a bit more complex. We followed published methods. And you can see here that there is potential that this could be useful. I mean, you for example, we find common SNPs to products from the same manufacturer at similar positions with different similar levels of mutations. So again, as mentioned, I think this is worth further evaluating. And this, thank you very much for all the collaborators, colleagues in my institute, other collaborators, and mainly Mansi Majumdar for working with me on this project. Thanks a lot.
Thanks a lot, Javier. If you can stay online a bit, we have a question into the Q&A box. If it's okay with you for me to read it.
Yeah.
So what is known about the cooperativity of OPV mutation with respect to neurovirulence? Is it, yeah, please go.
Yeah, as mentioned, there are some, there is mainly one mutation in the 5' that is the dominant mutation response for alterations. So if you have that, you kind of obscure other mutations, but definitely there are mutations that can accumulate and increase neurovirulence to each other. Yeah, and for example, we developed a novel vaccine called novel OPV, in which we modify the genetic profile, and in fact, it's much more genetically stable than OPV, but it can also accumulate several mutations, and with new mutations accumulating, eventually you can reach a high level of virulence. So yes, mutations can cooperate.
Mm-hmm. And the next piece of the question that we have online is that would longer read sequencing be an improvement compared to the short read sequencing?
Yes, I mean, I think there is potential that nanopore or long read sequencing could be useful because you have actually sequenced whole genomes, so you can actually identify how mutations map together. But although things are improving very fast, the sequence error of nanopore is still higher than those using short read sequencing like Illumina. It's approaching, so we are working on that and see how it compares, but I think we're still not there.
Thank you, Javier, and thank you very much for the question online. So there is still some work ongoing in order to really leverage at its best the technology of the HTS and NGS with regards to the sequencing opportunity and this identifying the different sets of the mutation. Thank you very much, Javier. So okay, with this, I would like to introduce our next speakers. They are both from Biological E. They are Dr. Panduranga Rao, Valurpiri Rao, and Pradeep Das. They are going to share what has been their experience on the monkey neurovirulence testings of yellow fever and novel oral polio virus vaccines. So, Dr. Pradeep and Dr. Rao, I'm bringing up your presentation. You're welcome to come online.
Thank you. Thank you, Laura.
Let me know when you see your presentation. Just a second, it's coming up. There it is.
Yes. Hi, everyone. Myself, Dr. Pradeep, and with me, my colleague, Dr. Panduranga Rao is there. Currently, we are talking on the subject on monkey neural testing of yellow fever vaccine and NOP and its refinement and replacement opportunities. So coming to the first slide is, as we know, the neurovirulence current status is, it is currently accepted in vivo safety and consistency for neurotropic live attenuated vaccines, which is classically for polio as well as NOPV and yellow fever vaccines. And this test is mainly used for attenuation of seed, work, and production lots before the release. So this is one of the mandatory tests. And for the new NOPV2 vaccine, which is newly genetically stabilized vaccine, monkey neurovirulence test and its procedures of development and manufacture to confirm the attenuated seed and lots produced during this NOPV2 vaccine. And still in the DS manufacturing, each bulk should be tested for MNVT, either by monkey or as well as transgenic mice, which we'll focus in our later part of the slide. Next slide, please. So coming to the main problems of MNVT, we think as a manufacturers which requires a complete replacement of monkey neurovirulence test. And as my other colleagues from Sanofi and Javier Martin has suggested, that is practically ethical and practical concerns, because every batch, if it needs to be tested on the non-human primates, and it has associated with its own welfare, pain, cost, and biosafety requirements. And biological variability and limited predictiveness. This is basically if you do some studies in interlaboratory variabilities and human adverse event, which is more the correlations, and we cannot correlate much more to the human welfare. So suppose mouse monkey neurovirulence test fails, it does not indicate that really it is your product is having a serious issue. However, we have to again repeat the assay, and it has basically having limited predictiveness. And as we know, currently most of the regulators as well as the local regulators are now giving lot of emphasis on reduction or replacement of use of animals. And currently the recent WHO draft guidelines also suggested the new technologies available for both this test, yellow fever and NOPV. So next slide, please. So my colleague, Dr. Panduranga Rao, will go over to the NOPV part.
Yeah, we are working with NOPV, and yeah, these whatever the alternative tests available, like transgenic mouse, MapRec, and NGS, that's already discussed by our colleagues. And yeah, one thing I want to add here is MapRec is not a suitable assay for NOPVs. So we don't have any other option but to go with the NGS. So and we have for all the batches, all the DS batches, what we are producing at present for NOPV2, we are using NGS as well as monkey neurovirulence. And mouse neurovirulence is available, but we haven't tested it. Our colleagues, other colleagues will be having a better idea about this mouse neurovirulence. But only one thing I see there is, in monkey neurovirulence testing, all I need to go is with histopathology and other things. Whereas in the mouse neurovirulence, clinical, this one should be enough. That will be a positive thing for me to discuss about my yellow fever vaccine testings using mouse. Can we go to the next? Yeah, coming to the alternative opportunities available for yellow fever. At present, there is only one test that is available, is monkey neurovirulence, but it is limited to banks only. It is not a release test for the batches, for the DS batches. It is only master bank and working seed, both we need to test, and there is a reference virus available. But during our development of a new yellow fever vaccine based upon cell cultures, we thought, okay, we will look into the mouse neurovirulence also. It is not a new test. This was potency test earlier, this one, before international units are introduced. This is the test that is being used for the potency. So basically 1000 LD50 is requirement per single human dose. That's what it is. And we thought, okay, let us see how it can be, whether it can differentiate different viruses of different neurotropism. So we have taken, that is one, the data will come later. And coming to the viscerotropism and immunogenicity, already Carey has shown that there are some, in hamsters and other things, viscerotropism can be done. And we also have seen that in rats, hamsters, and rabbits, immunogenicity is clear, but sometimes we see rabbits are better models than for the immunogenicity. Viscerotropism, we don't have much information about that. And coming to the NGS and deep sequencing, we have done sequencing of the at least three master bank, working bank, and the production batch, which we used in the PCT. Both NGS and monkey neurovirulence is done. And in NGS, we didn't see much polymorphism in our batches. In our banks, there is no polymorphism, probably because we purified our banks. Whereas monkey neurovirulence, when we looked at that, when compared to the one of the egg-based vaccine available to us, the NABS standard and our batches are slightly more safer or less neurovirulent or neurotropic. Other than that, we are not seeing much this one, and probably to establish this one, we need more collaborative studies. And coming to NGS, there is, as already mentioned earlier, the molecular determinants of attenuation is not yet very clear. As per WHO and the available literature, there are 24 potential sites, which all may not be potentially attenuating sites. It may be due to polymorphism. And these 24 mutations are seen in the 17D viruses compared to the ACB virus. So out of this, probably these are the important attenuation sites may be present in these 24 only, limited to these 24 only. So we have this one. Yes, we know some sites which is potential to have attenuation sites, but we need to test them, and all for that collaborative studies are required. That's where MHRA, WHO and manufacturers may compare. Next, please. Yeah, what we did, this is the mouse neurovirus study we have done with WHO 168-73 and our master seed, working seed, and one of the competitors' vaccine, and a placebo. This is simply no virus. So we have seen that this mouse test is able to differentiate this one from anywhere between 10 to almost 300 IUs. Okay, in our case, our master and working banks are more than 300 IUs. We could not see any deaths. LD50 is more than 300, whereas our batch used for the PCT, that is around 144, whereas in the competitors' vaccine, it is around 10, and 168-73 is also around 10. So this shows that this can differentiate this type of differentiation we could not see in the monkey neurovirulence. So probably mouse, we need to do these tests again and again and see whether this can be a good model as a replacement for monkey testing. Next, please. Yeah, this is already discussed, and these are the 24 potential attenuation sites, which are different from means the differences between CB and the 17D viruses that are being used. And these are the NGS we are looking at for all the batches. We are looking at NGS for all these tests. None of them are more than 1% in our batches. Next, please. Yeah, this is the summary. Clearly, yellow fever and OPV, when I compare, in yellow fever and OPV, master and working seeds need to be tested, whereas the drug sufficiency is not required for yellow fever. That is a positive thing. Whereas when we are doing the primate testing, we need to do viscerotropism, immunogenicity, and neurotropism in case of yellow fever, whereas in OPV, a neurovirulence test only is present. And for immunogenicity, viscerotropism, and neurovirulence, there are alternative small animals available. So we need to look into that. And for both, reference viruses are available. WHO reference viruses are available for both. And the most important part is containment requirements. In case of OPV vaccines, even for the animal testing, we need GAP4. So even for mouse testing, it requires GAP4. So we need to more move towards the NGS, whereas in case of a yellow fever, a BSL2 may be enough. And in case of yellow fever, determinants of attenuation is not known, but OPV, it is very well known. And there is only one primary attenuation site, though other attenuation sites may be present. But one of the recommendations by WHO is moving towards the HTS for OPV, but not sufficient data is available. But we believe if the genotype is similar for the known non-neurovirulent strain, that HTS may be used as a regular testing. And coming to refinement, yellow fever, this one, there is a potential that other smaller small mammals can be used for viscerotropism, immunogenicity, and neurotropism instead of monkeys. That opportunity is clearly visible, and whereas OPV, it is already available, that is TG mouse. So this is with this, what I think is we should move towards with the success of OPV testings, coming to the refinement of going to mouse and other things, and now with the lot of data and HTS, now it is time to move to yellow fever also, to refine this one, going to the lower mammals and going with the NGS. That is the need of the hour, we feel. Thank you.
Thank you, everyone.
Thank you very much, Dr. Rao and Dr. Pradeep, for your presentation. Let me see if there are questions into the chat or into the Q&A box. It doesn't look like. Okay, so thank you very much for your presentation and sharing what has been your experience so far for both yellow fever, OPV, and the new experience with the novel oral polio virus vaccine. So thank you very much. We move to our next speaker, that is Dr. Tong from Health Canada. Dr. Tong, thank you very much for being with us. I'm bringing up your presentation in a second.
Thank you.
Let me see let me know when you can see it. Please, the floor is yours. Thank you.
Yes, thank you, Laura. And I also want to first thank all the presenters that highlight those technical challenges and the scientific informations. And this actually make my job to walk you through some of the scientific and the regulatory considerations related to controlling of residue neurovirulence of viral vaccine are much easier. Next slide, please. Because I am a regulator, I just wanted to mention that the presentation is my personal opinion, not the official position of my agency. Next one. And as you all know that live viral vaccine have been used successfully for the control of human disease. Some of the best known human vaccines are in this category. For example, smallpox vaccine and OPV that has been discussed extensively in this webinar. And of course, because live vaccine containing a live virus, it is critical to ensure that the live virus are adequately attenuated, and the attenuation is maintained for each commercial lot. And the neurovirulence should always be adequately studied when you're developing a new live viral vaccine prior to a clinical study, and the residue neurovirulence of the production strain is important to be studied, and to show that the residue neurovirulence is adequately controlled prior to proceed to inoculating those products into humans. For example, some of the virus that we know that they have neurovirulence properties, such as polio virus, mumps virus, and other virus, the attenuation is achieved through serial passages in tissues of central nervous system. So those kind of situations, neurovirulence study need to be part of the preclinical data package. Of course, when we move to routine manufacturing, neurovirulence tests are performed in case of not unacceptable lot-to-lot variability in residual neurovirulence. I think the best example is really OPV, that each drug substance monovalent virus are tested. And the monkey neurovirulence test is the best known assay, which has played an important role in ensuring the safety of several live viral vaccines since the early 1900s. Next, please. Here is a summary of required neurovirulence tests specified in WHO document that published prior to 2025. And it is a high level of summary. And I think the best known neurovirulence test is OPV. And the current guidance document specify that both sapien OPV and as well as novel OPV neurovirulence testing should be conducted at virus seed level as well as each monovalent bulk. And the test could be high throughput sequence or in vivo assay in monkeys or in transgenic mice. And the Japanese encephalitis vaccines also require neurovirulence assessment for virus seeds in mice or in monkeys. And each final bulk or final product should also be tested in mice. And the new vaccine for dengue, although there was no specific neurovirulence assessment for dengue virus, however, if the antigen is used yellow fever virus as a vector, and the test for virus seeds should be conducted similar to yellow fever viral vaccine itself. And also for measles, mumps, rubella, yellow fever, and the varicella vaccine, and the test of virus seeds in monkeys is currently required. And the last one is a smallpox vaccine, which is not attenuated vaccine, but it contains a live virus. And the test in mice in some situations can be in rabbit, is required for virus seeds. And each bulk suspension and should also be tested similarly as the virus seeds. I think this is a very high level of summary for the current product-specific requirement. Next one, please. I just want to, at a very high level, to highlight the in vivo assay that we performed for controlling neurovirulence, residue neurovirulence are not perfect. Okay, so first we're looking at the yellow fever vaccine, which has been discussed in detail. And I think it's already included. Here is just a very simple summary. In 1941, cases of encephalitis were reported in individuals who received yellow fever vaccine prepared from a 17D derived substrain. And a subsequent study revealed that different 17D derived substrains of yellow fever virus production material that have different level of residue neurovirulence. And because of this discovery that a seed lot system of the production strain was introduced, which is a cornerstone of the manufacturing control for all biological products today, and greatly improved lot-to-lot consistency. Of course, along the way, monkey neurovirulence test was also introduced for the control of yellow fever virus seeds, and the assay has been improved throughout the time. However, even with all this manufacturing improvement, yellow fever vaccine is still associated with very rare neurotropic disease. I think from all of this, we can say that monkey neurovirulence test for the control of yellow fever vaccine residue neurovirulence is very useful because they can actually distinguish different level of residue neurovirulence, but still itself cannot eliminate rare yellow fever vaccine associated adverse reaction. Next one. Okay, I think OPV, the control of OPV have been discussed by Kostya and Javier. And I just want to also highlight it, as both of them mentioned, the monkey neurovirulence test is based on the correlation and based on the consistency of the product. And it's not absolute prediction. And here, a few examples, and some of them already mentioned by Kostya. First, in 1960s, the Institute of Sera and Vaccines in Prague, they developed a type 3 attenuated OPV. Judged by the results of monkey neurovirulence test, this new OPV3 strain actually is expected to be at least as safe or even safer than Sabin type 3 Sabin strain. However, the clinical study of this type 3 new type 3 OPV led to an extensive outbreak of vaccine-related poliomyelitis in 1968. And also published data review 80 Sabin type 3 lots manufactured between 1964 and 1983 in the US. And the type 3 OPV is less neurovirulent than the type 1 based on the monkey neurovirulence results. However, the failed use results shows that the type 3 OPV is associated with vaccine-related poliomyelitis actually more frequently than the type 1 OPV. And I think another is already mentioned by Kostya, those partially attenuated poliovirus as determined by intracerebral and spinal inoculation in monkeys, but they are not paralytic at maximum dose in chimps. So those experiences highlighted that monkey neurovirulence tests are not always predictive of residual neurovirulence in humans. And I think the way it is used for the control of OPV is based on correlation and based on the consistency of the product that we between the commercial lots and all the lots that we have showed safe, acceptable safety profile in clinical study or in general use. Next one, please. And the other example is mumps vaccine. Actually, monkey neurovirulence test didn't work so well for the detecting of residual neurovirulence of the live mum virus vaccine. And there was an attenuated mumps strain called Ubra M9. And this strain passed the monkey neurovirulence test and was subsequently licensed and to produce a commercial product. But the post-licensure experience that with this particular commercial product, a number of septic meningitis cases associated with this product was reported in Canada, Japan, and Europe in the late 1980s. Now, this strain-derived live mum vaccine are no longer marketed. And later on, because the deficiency with in terms of predicting the residual neurovirulence and the more sensitive test using neonatal rats was developed, and which can better predict differentiate, attenuate different level of attenuation associated with mumps virus. And I think this experience also highlighted monkey neurovirulence test may not be a suitable animal model for all live attenuated live virus in terms of predicting residual neurovirulence. And I think the neurovirulence pathological manifestation of this virus in monkeys may not always correlate because it doesn't correlate to the neurovirulence in humans. Next one. And I think overall experience is very consistent with my colleagues just discussed related OPV and yellow fever vaccine. Monkey neuron test was used for almost a century because this is our best option at this time. And we have limited understanding concerning the mechanisms and the genetic basis of attenuation in the past. And of course, we don't have the technology we have today, such as the sequencing. And I think we need to understand the interpretation of monkey neurovirulence test is largely based on the correlation between the neuropathogenical manifestation of the virus in monkeys and the strands known neurovirulence in human. And I think this is the basis moving forward for the control of commercial products if the test is used. And we have to recognize that this monkey test is not suitable for the control of several viruses. I think this is mumps for, yes, we already knew, and I think it's also recognized may not be suitable for measles as well. And even with this test in place, and they cannot eliminate rare adverse reaction that is associated with yellow fever vaccine and OPV. These are just part of the characteristics of the vaccine itself. Of course, advances in molecular biology have led to new methods for neurovirulence assessment based on improved understanding of mechanisms and the genetic basis of attenuation, and more sensitive technologies such as NGS is currently available. Next one. And I think I just want to highlight a few advances for OPV control, which has already been discussed. I think it's just for the sake of discussion. And the genetic basis of attenuation, of course, for the both for the attenuation and the reversion were identified for OPV. And of course, in the 1990s, Marburg test was developed by FDA, CBER FDA. And also a neurovirulence test in transgenic mice also developed. And as my colleague has already mentioned, currently high-throughput sequence become available. Actually, some manufacturer has already 5-year experience, more than 5-year experience with HTS for the control of neurovirulence assay. Next one. So, as we always mentioned, or we have mentioned already that consistency approach is the way of ensuring safety and the effectiveness of a commercial vaccine product. And what is the regulatory expectation after you license a new vaccine? Of course, commercial vaccine lots, they should retain the safety profile as well as the effectiveness or efficacy that are demonstrated in successful clinical studies. And because we no longer doing any clinical study post-licensure, and assure those quality of the assure those safety and the effectiveness of the vaccine is entirely based on the control of quality of the product, and the guiding principle is based on the consistency principle, which is the quality attributes of the commercial lots throughout product life cycles. They should be comparable to those of lots shown to be safe and effective in clinical studies. I think this is the basis for the control of commercial product. And I think this is, the concept is different from, let's say, in the 50s, largely rely on the testing of the for release. Today, the quality of commercial vaccine lots is built into manufacturing process. We have stringent, good manufacturing practice, and we also have adequate control of starting and raw materials. And the manufacturer, they are using validated manufacturing process. They implement robust and appropriate quality control steps, control tests at appropriate manufacturing stage. And also, and the other important aspect is implementing appropriate quality specifications. Next one. And here is kind of a proposal of a strategy how to control neurovirulence and when we're developing new live viral vaccines. Of course, before you even start, you have to collect all the scientific information and all the scientific study, and to have assessment, is neurovirulence even associated with this particular virus? If the answer is yes, I think the preclinical stage, there should have some studies to address that, to assess. And I think only when you have reasonable confidence that the residual neurovirulence is at a level safe for human use, and you can move forward to initiate your clinical study. And throughout the clinical study, I think it's important to start to generating quality information, which the most important one is the virus sequence. And I think if residual neurovirulence levels throughout the development stage are considered consistent and safe in humans, when entering a commercial production, there was no need to do testing at each lot basis. However, I think it is always, given the technology available to us today, I think genome sequencing probably is a very important control for the viral C lots. And of course, if a lot-to-lot residual neurovirulence level vary significantly, as similar to what we see in OPV, and I think in that case, the residual neurovirulence need to be tested and controlled at each lot level. But this can be done actually through a clinical, through the genetic profile that he already collected through clinical development. Next one. As a colleague already mentioned, WHO has been last year drafting a guidance document on the replacement or removal of animal tests for the quality control of biological product. And of course, neurovirulence test is covered in this guidance document. I just wanted to give you a rundown of the status of this guidance document. The final public consultation was completed in September, and the new guidance document was discussed and adopted at the 81st meeting of the WHO Expert Committee on Biological Standardization just last week. And I think I want to highlight some general considerations on why we even move forward. Actually, 3R is not the driving force to move for this guidance document. It is only mentioned once in the as a historical piece, because I think the guidance document recognizes the scientific limitations of many animal assays, particularly in the context of routine batch release, because animal-based assays are highly variable and sometimes lead to more frequent both invalid tests and failed tests. And the failed test is not because of the issue with product quality, often is because of assay variability. And I think the guidance document recommends the use of scientifically relevant in vitro methods over in vivo methods for routine quality control of biological product. And the recommendations in this new guidance document will be supersede any corresponding quality control requirements concerning in vivo assays specified in WHO guidance document published prior to 2025. I think this going back to, I have a table of summary of product-specific requirement concerning neurovirulence test. And the new guidance document will be supersede all the product-specific method. Next one, please. And specific consideration for neurovirulence testing in this guidance document is very much aligned with the propose the strategy I highlighted earlier, which I actually discussed in other meetings a few years back. And I think that at the non-clinical stage is recommending the same kind of principles. You assess the if you are developing a new attenuated viral vaccines, whether the need to perform neurovirulence, it should be based on all available scientific informations. And if it's needed, and we should select suitable animal models. I think this is also, of course, we have a lot of experience with monkey neurovirulence test for polio, OPV, as well for yellow fever. But if you select other animal models, you should provide information to demonstrate the suitability of the animal models. And I think, and this is always, and then the other ones, I think because advance in technology and many researchers are developing in vitro system, those should be considered only when demonstrate its fitness for use. Okay, next one, please. Of course, at the clinical development stage, when you are testing materials in a clinical, it's highly recommended that genome sequence data are generated, because this is going to be the foundation for moving forward using in vitro assay for commercial production. And of course, for commercial production, residual neurovirulence is controlled at appropriate manufacturing steps. This it depends. If the virus is stable, there was no need for conducting those type of testing for on batch level, and of course use appropriate molecular method. I think this guidance document is very, very high level in terms of principle. It did not provide how you the detail how you're going to which method to select, how you validate and control the assay. And I think this is my last slide, but I think I also recognize moving forward to use a sequence-based assay for lot release or the control of viral seeds. And there are still a number of challenges. And for example, the control of the assay, the validation of the assay, I think most importantly is the specification. Of course, as we mentioned, the approach is consistency, and the commercial laws should be consistent in terms of genetic profile that in comparison to clinical laws that you have shown to be safe and efficacious in clinical studies. And for some legacy product, such as yellow fever, and you can also going back to looking at the commercial product that have been used with no concerns about the safety, and those data could also provide additional support in terms of setting specifications. I think this is all I have to say at this point, but I think we need to continue to discuss the challenges for implementing in vitro assays. Over. And yeah, this is my colleagues contribute to the topic.
Thank you very much, Tong, for your presentation, and also for sharing the very good news about the approval of the WHO guidelines. This is going to be a very fundamental document that hopefully is going to guide all the stakeholders worldwide into their planning to transition to non-animal testing, although the monkey neurovirulence test might be one of the challenges that still need to be kind of tackled in terms of implementing the HTS or NGS for the live viral vaccines as well. So thank you very much. And we still have some time together, and I would like to welcome back all the speakers of the webinar. And I would like also to introduce Dr. Kutub Mahmoud from PATH, who is going to be the moderator of this panel discussion. And again, I would like to encourage you all to add any questions into the Q&A. It will be very important for us again to hear from you. If you have questions, doubts, concerns about what you have been listening since a couple of hours with regards to the implementation of those new technologies. Before giving the floor to Kutub, I would like to remind the people online to fill in our survey, because we would like really to better understand your experience with the monkey neurovirulence test and the current available replacement opportunities. So again, Kutub, the floor is yours. Please, all the speakers turn on your camera. We hope to have a very nice discussion together about the next steps. Please, Kutub, thank you.
Yeah, thank you, Laura. Thanks all the speakers. This was very informative session related to the use of the high throughput sequencing in replacing the neurovirulence testing. As a next step, we have seen so many guidance documents. We have seen so many publications around the use of the HTS. I think what it comes down to the implementation of the HTS, and what would be, I think, the questions that will come up is what is needed at the ground level in terms of the implementation of the HTS. This has been the challenge, and we have seen 2025 this year particularly. There have been quite a few webinars, and including this one, and we have had good attendance from manufacturers, regulators, and all. But at the implementation level, it has been only few regulatory bodies that are implementing this. I think these are some of the things that I came across. But as next steps, I would like to hear from the speakers, what would it take to have this more and more improved. I'm sure there will be progress in the years in terms of implementation, but are there any specific things that each of the speakers recommend? Because we now have all these guidance documents from WHO, from EMA, and other guidance documents. But implementation is one thing. I thought I'd touch on this. So with that, I would like to maybe go back to Tong, and then she can pass it on to the other speakers from her experience as a regulatory body in terms of the implementation. Over to Tong.
Thank you, Kutub. I think I'm going to just outline what I think, but not for my agency, as personally what I think. I think we have two categories of situation when it comes to neurovirulence test for the commercial production. One is like OPV or NOPV, because NOPV in particular, it is a designed sequence to improve the stability, and we have 5 years experience. And I think in those cases, we know where the few nucleotide, those sequences are so important in terms of residue and neurovirulence. So in those cases, even we use high throughput sequence to measure each lot, maybe is the past failure. Should we, personally, I think we should be focusing on those important area, such just a few nucleotide sequence. And I think set the specification. So this is one for some well-studied, where we know the mechanism, where we know exactly, well, I think we have a good idea where they are. And I think looking back for all the commercial laws, you have generating quite a few years of data. And if this could lead to good not release decisions, let's say, because of course you can do the entire genome sequence and set and ask for the require consistency of entire genome. But I think this is also coming back to the notion that with more technology become available, and those are provide a lot more information. And I think we are replacing neurovirulence test, in vivo neurovirulence test. We should focus on those relevant to neurovirulence, because I think otherwise we may put a lot more burden on the manufacturing side. And of course, if you already realize with your experience some of the change will affect immunogenicity, and you should be added to it. But that is related to immunogenicity because we are replacing neurovirulence tests. But I think yellow fever is a different story because we don't know the precise change need to attenuation. And in that case, the consistency is the only way to approach the assay replacement. However, I think we already know that there was no need to conduct those type of tests previously on each batch. So whatever we set the limit for the virus seed lot control should also be accordingly, because they will be using, if you're using an NGS, you will be coming out with more information. That means, single nucleotide polymorphism, you will be detecting those. And we should not be control things that maybe in the old days, we don't control is perfectly safe. I think this is where I'm coming from, because if we get a lot more information, we want to tight control, and it's not going to be incentive for the manufacturing moving forward. I think, but of course, in any means, we should never kind of relax the control in terms of residual neurovirulence. This is kind of my thinking. Over.
Yeah, thanks, Tong. Yeah, for NOPV, we do have the whole genome sequence data. So not only we are addressing the neurovirulence, we also see whether the immunogenicity antigenic sites are retained and are those impacted. And that also we have done for other S19 IPV development also. Now, I think from the manufacturer side, we have seen some challenges where if the high throughput sequencing is contracted out, because if they don't have the equipment or the facilities or trained personnel in the manufacturing unit, they outsource the testing to a CRO where they do the sequencing. But at some point, the regulators or the WHO pre-qualification team, they would want the test to be brought in-house for them to have this assay running. Is that a challenge? So maybe I would like to hear from the vaccine manufacturers. We have several here. So maybe coming from the LMIC or from India, maybe BioE can address this. What are the challenges? Outsourcing the high throughput sequencing when we don't have in-house high throughput sequencing, or bringing those to comply. Are there any regulatory requirements or WHO PQ requirements to have it in-house? Over to you.
Thanks, Kutub. I think from the manufacturing perspective, we have the NGS, but the main things for the replacement and also the CRO we have to do the studies, but main things for the replacement and the correlation between monkey neurovirulence test and NGS, that platform needs to be a little bit defined, what exactly needed. So it also maybe also dependent case to case, regulators to regulators, what are the things we should do because NGS is very, very vast, and to validate it, it is a cumbersome, tough task for the manufacturers without any particular guidelines. Because guidelines just say you can use any validated HTS methodology, but it may have different analytical variabilities which needs to be fixed in some part of a documentation or in the compendia. So that is the very key challenges we have. And with one time we can outsource this test, but as you know, there are multiple viral vaccines coming together. It is every time outsourcing this job and sending the samples to the outside countries. It is always a tough task by so much of hindrance related to customs, clearance, their NOC requirement by other countries. So definitely it is a tough task. And as a BioE, we have now NGS with us, and for every viral vaccines, we are now getting the genetic sequence done with the help of PATH. And thanks you for your support to BioE. Dr. Pandu?
Yeah. Coming to this NGS, the major challenge I see from manufacturing this one is we can purchase the equipment and we can get the manpower. That's not the big issue. The big issue is reference reagents. And whenever we are going to the outsourcing, the big problem is containment. We have several NGS means outsourcing agencies available in India, maybe 10 or more than that. But most of them, they cannot handle the virus. Okay. So here this one is, if we can get the reference reagents, we can do a in-house so that the containment issues are cleared. Otherwise, we need to develop some these outsourcing agencies where they can have containment. But I don't think that as a viable solution, and reference reagents and in-house development is the best way to go at key.
Yeah. Pandu, with respect to the Indian regulatory authority interactions, do they also do the HTS, or they review the data, or what is on their side to review, to implement HTS in vaccine release?
Yeah, HTS, this one is new to our regulators also. Okay, so recently with the help of you and PATH, we conducted a training program for the regulators in BioE for them to understand what is exactly is done and how to interpret the data, how to use the bioinformatic pipelines and other things. Thanks to MHRA and PATH and other people, probably that is what is important for, at least for the manufacturers in India and other developing countries. Means for the regulatory also, we need to consider, we need to conduct the training programs for them also, so that they will be more comfortable reviewing our data.
Okay. Thanks. I would like to hear from other manufacturers. We have Sanofi here. Karim, maybe you can, I'm sure you guys may be implementing this in the company, and you face different regulatory authorities globally with your products. How do you deal with implementation of the high throughput sequencing across different vaccine platforms?
Indeed, luckily we have internal NGS capacity, both long read and short read. I think the challenge to anyone who wants to use HTS for yellow fever will be how to define a threshold that is both scientifically relevant and that is that suit the validation and that is useful in regards to the neurovirulence. How low shall we be in our ability to detect a real variant that may be present? And that's why, to me, for the moment, it's good to still have the evaluation within an overall testing package, not only based on the sequence, but also on the plaque size, any other phenotypic assessment you can do within your viral population. Because HTS is wonderful, but if we are looking for variants that are at 1%, how relevant is those? Are we able to get those 1% subpopulation? Are they relevant, or is it just a sequencing artifact? And can we validate that they are at 1%? And who will define that the threshold should be at 10% or 5% or even below? So that's maybe the difficulty with implementing the NGS alone for assessing the neurovirulence or the consistency of yellow fever vaccine when we need to be able to define a threshold that is both scientifically relevant in regard to the vaccine, but also technically relevant on what we can expect from this technique.
Yeah, I think you raised a very good point. At least for polio, we do have this 1% cutoff for the mutant in the domain 5, but where we do have a genetic marker for attenuation, and for other vaccines, it may be a challenge. Yeah, from regulators, I would like to hear maybe Kasia, you can open up from the regulatory perspective how the HTS you see going forward. Over.
No, I just want to respond to Karina's question about the 1% and how do we go about determining this threshold. I think it's, first of all, I mean, we all understand that we're now only talking about the routine lot release, not about the developmental phase when we don't really know much about the product. Once you establish, and at that first phase, of course, animal testing would perhaps, sadly, will remain relevant. But once you establish your production and you accumulate data on the S&P profiles, then you just basically follow consistency model. You determine whether a new batch is significantly different from the batches that were tested in all irrelevant biological tests. So that's, I think, the only realistic approach, because, you know, interpreting the significance of any molecular signature that we detect is, I mean, at least at this point, it's not possible. We don't really have a good understanding of how genetic, you know, markers relate to phenotype, with very few exceptions. And a polio vaccine, I mean, we were extremely lucky that it was able to determine, to identify almost a single or just very few molecular mutations, you know, nucleotide polymorphism that are related to this important characteristic. For other vaccines, that may not be possible, I mean, in the future. So I would suggest that we rely exclusively on this consistency model. And as to the regulatory aspects of this, definitely new products, and we must also understand that this approach is relevant not only for live viral vaccines, not only for oral polio vaccine or yellow fever or mumps or whatever. It's relevant to any product that is manufactured by growing something, like, for instance, gene therapy vectors, oncotherapies based on some, you know, viruses and so on. So this is broadly applicable to a range of biological products. And for those new products, I think it's a no-brainer. I mean, this should be used and will be used for manufacturers or developers of the new products, biological products. I mean, they probably will jump on this opportunity once this is a kind of a test case, and we hope that OPV will be kind of a test case. And if we get through ECBS, this recommendation, then it will open the gates for other products. But for the products that are the old legacy products, I think, of course, it will be a little more challenging, because as we all know, replacing the existing techniques that are prescribed in the license, this will be not only would potentially require some general guidance from the World Health Organization, but also it will be determined by the national regulatory authorities. So I envision that introduction of this new approach will be more challenging for the existing products. Over.
Yeah, thanks. Thanks, Kasia.
And if I may, I'm perfectly aligned, and I agree that the consistency is the way to go. What I wanted to highlight is how do we demonstrate that is equivalent to the reference material, and it's how we define the equivalence. That's the problem we face with the clinical scoring within the past test in non-human primate that we executed, because it was because of the statistical analysis that we failed, while we know that the material is perfectly fine and that at the sequence level it's perfectly equivalent. So it's how we define the tools to say, yes, it's the same material, it is consistent. And where we put the bar to define this equivalence.
Well, look, I mean, this can be done statistically, and as I demonstrated in my presentation, I mean, it is possible for polio virus, and I see no reason why it shouldn't be applicable the same approach for any other product. First off, you look at the nucleotide polymorphism. You accumulate a historical database of all possible variations. You determine the range of what's feasible to achieve, the consistency level, the range of possible variations. And then you just keep on updating this database by adding new lots that you will produce down the road. So that's, I think, is a way to go, essentially starting from what you can achieve. Because, I mean, different products perhaps will have different variability in terms of their genetic composition. Some products will be more tight, and I would actually look at this approach more like a way to raise red flags. You suppose you don't have this approach. I mean, you will still be making vaccine lots and you will still be releasing them, right? But you will be in the dark. You will not know what is inside. I mean, what is the genetic signature of your product. But now you will see it, and all of a sudden you have something that is not consistent with what you were able to produce before. So for you, it's a kind of a signal that you need to do some troubleshooting and understand what's going on. In most cases, it will mean nothing. It will be just, okay, I mean, it's different, so what? But in some cases, maybe it will signal that you should really control your manufacturing process more tightly.
Tom?
I think Karen and Kasia, I think the three of us are absolutely aligned on this one. Of course, Karen, it's a little bit difficult to be specific, but I think you do have, I think the principle is we line up with whatever tested clinical study, but yellow fever is a legacy vaccine. But looking back, you have produced so many commercial lots that are completely have the acceptable safety profile, and which you never even assess residual neurovirulence. But you can go back because those are the baseline you can, based on your future decision on, you can actually generating NGS for those material. And if there was one particular nucleotide that has been with a mixture of 20% of something else, and in few lots never had a problem, and this is your acceptable limit for it, you don't need to control it at 1%, right? And I actually doubt any of them you need to control at that level. OPV is a very special case because those mixtured kind of genome is always present in any kind of each manufacturing lots, and because probably it's because slight pressure. Over.
Yeah, just one more, comment to confirm what Tong was saying, because even for polio, for which this marker of neurovirulence was established, this type 3 is a special case. It's a remarkable thing that 1%, just 1% of a mutant can produce such an effect when looked at in the monkey test. For type 2 and type 1, it's very different. So it's not until you reach 10, 20, 30% of the content of this highly virulent mutation, then you can see something in the monkey test. So I think that 1% is, we chose 1% for this just to kind of to get rid of the background noise. It's a noise. And also another way to look at what is your noise level is to see how your NGS protocol in your particular lab performs. And for this, actually, the reference materials that will be provided for polio vaccine, at least, and I think it should be done for other products as well. It will give you the baseline level of the noise, just technical, variations that are inevitable in any lab procedure, and NGS is not an exception.
Thanks, Kosya. I just wanted to also hear from MHRA if there is any one institution that has done a lot of neurovirulence testing and training other institution. It has been the NIBSC, formerly NIBSC, now MHRA. They have been the pillar for doing the neurovirulence testing. I would like to hear from MHRA with respect to the implementation of HTS now, moving away from neurovirulence testing, and what as a regulatory body you see some challenges to MHRA. Javier is there, I think, and anybody else?
Let's see, Kutu, because Javier might have had to jump on another meeting.
Okay.
Let's see if he's able to reconnect.
I think Manasi is still there. Maybe you can put her in this. She's in the attendees list.
And
thank you.
Maybe while we wait for Blaise, do you have any comments on from your side?
But I think that the specificity of polio vaccine is that it's a testing that we do on each and every bug, differently from other mumps, measles, and others. Can you hear me? Yeah. And so for that, of course, we have a large amount of knowledge, of database in terms of monkey neurovirulence test, but also in transgenic mice now. So this makes it super rich in terms of database that we can connect to, which may be a little bit different from others where you do only the seeds. And maybe this introduced the idea of, okay, for this, you need to release your seed lot. And I don't know how many seeds, but it's certainly less than the number of release lot, bulk lot that you have. So it's a bit like we are more in the characterization world than release on a batch release each and every lot. So I like in the presentation of Karine the fact that there are indeed other characterization tests, and we are speaking about that in order to highlight what I took in my notes. Let's go back here, the biological significance, because this is the key, basically, the biological significance of what we see in terms of changes in mutation. What does it mean at the end? And even that threshold where we stand, what does it mean exactly biologically? And One thing maybe we can come back a little bit on, and I have more question on that, is the capacity of those viruses to cross the hemato-meningeal barrier. So the mini brain and the organ on a chip, basically, maybe an interesting tool also for characterization and to elucidate in a way this biological significance of what we see in terms of mutation.
Yeah, thanks. I think what we have heard is consistency is also very important from Tong, batch-to-batch consistency with the high-throughput. And then you raised a very good point about the biological activity. What is the relevance of this data that we get from the high-throughput sequencing? All this in the context of neurovirulence testing and replacing that with high-throughput sequencing. I think these are all the key points. I see that maybe next year we will have more progress. There's already recommendation by EMA to implement high-throughput sequencing for adventitious agent testing starting from April of 2026. So there will be a time when there will be a recommendation for the implementation of HTS for replacing neuro or not only completely replacing them, because we still need some testing for the seeds, but there will be at least implementation of HTS for vaccine lot release. Any other speaker have any comments before I hand over to Laura? Please. I didn't get Javier. Yeah, I see Kosia's hand raised. And then after Kosia, if I don't hear from Javier or Manasi, I'll give it back to Laura. Kosia.
Yes, I want to raise some unpleasant question, because I mean, we all hope that we will try to minimize, because just like Kutub said, it probably will be very difficult to get rid of animal testing altogether. But how do we go about maintaining proficiency? If the animal testing will be done only on an occasional basis, then it will be challenging to make sure that labs still know how to do it and can do it quite well. And I think that manufacturers and the regulatory authorities have to think about it, how we can maintain proficiency in the tests that we want to get rid of.
Yes. Absolutely, Kasia, very good point. I think training is one component of that, and then follow. I think Javier is back online, and he has had his hand raised. And I said MHRA have been the pillar on the animal testing, neural testing. So I'll give to Javier to comment.
Yeah, I believe there was a question for us, and Mansi just told me. So can you elaborate?
So you guys, MHRA, historically you have been doing neural testing for vaccine lot release, and for the HTS implementation. We just spoke about adventitious agent testing being implemented with HTS starting, say, April of 2026. When do you see that happening? Anything for some of the vaccines that we are doing where neural testing can be moved away, and then we have HTS? Over.
Well, I think that is not only for us, for you as well. It all depends how well we prepare this report and ECBS takes it and accepts it. But I guess after that, there might be more discussions with regulatory authorities and other stakeholders. It's a big step, isn't it?
Yeah.
Of course, a strong recommendation from ECBS would help a lot. And from what we've seen and we have discussed in many meetings, there is a lot of potential, and I believe this is a good test, and so far it's going very well. So, you know, within a year, hopefully.
Okay.
I mean, one of the first steps could be an OPV2. An OPV2, I think, is totally ready for this switch. Yeah.
Yeah,
So,
but then
Javier,
do we need a
sorry,
separate? Yeah. Go ahead, Tom. Separate document do we need for NOPV, or because if it is just one vaccine product that has progressed towards releasing with HTS, we would have to cover for OPV and IPV also.
I mean, we would probably need to change the TRS and explain how this needs to be conducted, and we could establish that, you know, a dossier for any particular vaccine can be prepared. I mean, another issue now is obviously testing is now possible in very, very few places, and that's why the monkey test remains necessary, because only very few institutions have implemented a transgenic mouse test.
Mm-hmm.
So that's why they haven't switched to that. At the moment, we used to do annual training. We are not doing that anymore. We have the problem with the PEF situation, containment. So, you know, there are many reasons for moving quickly.
Tom? Yeah.
Actually, Katuba, I think from ECPS point of view, we already allowed the use of NGS for neurovirulence assessment batch to batch for OPV and NOPV 2023. And this year, with the adoption of this new guidance document for any for any product, you can moving forward with this as long as your assay is controlled, your specification is approved by your regulator. Actually, Javier, we don't need any new document or approved by ECPS anymore, because this new guidance document for the replacement of animal assay with in vitro assay is supersede all the previous guidance document. So in principle, you don't need any
TRS.
Yes.
Not the collaborative
We don't
study.
need to revise anything.
No, but ECPS needs to approve our collaborative study.
That's a separate issue. That's a separate issue. And yes, you will provide better controls or reference reagents, but the company has some validation and has some experience, and its data is based on data. If it's consistent, you don't see anything, no detection of any kind of mutation you worried about. I mean, it's time to move forward. So
yeah, but you think any regulatory authority in any country has the capacity of reviewing that? Come on, you need some guidelines explaining what you need to look for and what are the thresholds, all these things, or not?
Well, we're doing that for a lot of things with no strict guideline. You have to based on the principle is out there is based on whatever you have demonstrated to be safe. And
yeah, normally when WHO approves guidelines, maybe there will be a workshop.
Yeah, that is very good. That's a good thing. Kadu. I mean, workshop, we continue to discuss more in technical details how to move forward. Can publish paper about this. Because there was no way WHO can do at this level. The Ivana's group has a lot of huge workload to manage. It's not able to do at this level. Yeah.
Yeah, but if I may, I think that the document that Tong is talking about, I think it is a great breakthrough. I mean, I think that it will open the possibility to apply to other products, but still, you know, some product-specific guidance would probably be necessary. I don't know in which form, TRS or a workshop, implementation, and perhaps even some research, you know, maybe perhaps even some collaborative studies. Because I think without specifics, it's just hand waving, you know, saying, oh, you can use NGS. So,
yeah, exactly.
Yeah, I think it's still important. There's a lot of things in front of us.
Yeah.
Yes.
But so Tong, are you in this WHO group?
drafting group, yes.
So have you discussed implementation workshop?
Actually, we did not, because there was no money for anything. You know, the huge cut, 40% cut in WHO budget.
You know, but that shouldn't affect the safety of vaccines.
Well, we have to find other means, right? So that's, yeah. I mean, it's challenging time for vaccines.
So, any other comment? Last comment from anybody, speakers or anyone else before I hand over to Laura? Going 1, 2, 3. Back to Laura. Yeah, to you.
Thank you, Kutub, and thank you very much to you all. I think that's there is much probably to keep discussing about this, especially about the implementation, especially about how we can maybe perhaps work all together to make sure that we support the implementation of those technologies. I think that's because of the complexity of the current situation, as Don highlighted, we probably really need to understand what kind of role everyone can have, starting from the companies that they do have access to an amount of data that is impressive in terms of the stability of their production line, about the consistency, and they have definitely a very important responsibility because of the amount of data and experience that they have with regards to their legacy products. So I know it's a huge step for them in terms of the amount of investment that they also have to do, especially if we think about the low and middle income countries manufacturers. This is probably not a very straightforward shift for them. At the same time, what we always recommend in all our discussion between manufacturers and regulators is really to make sure that discussions are taking place between manufacturers and local regulatory agencies. This type of dialogue is fundamental to build trust on the data that the companies are available about their process to implement novel technology. Could be the NGS or the NGS, but any other technologies with regards to the shift from in vivo to in vitro testing for the release and the stability of legacy products. So we definitely, as Animal-Free Safety Assessment Collaboration, we are actually happy to continue to leverage those type of dialogue, understand how we can maybe facilitate all this type of continuous conversation. And with that, I think that's myself and my colleague Jen are very happy to moving to the closure of these events. I thank you very much really for the time that you gave to us today, and in general during this year different workshop and webinars. Just a few closing words from Jen about the activities that we plan for the future. So thank you very much. Jen, you can close the webinar.
Thank you, Laura. So yes, I just wanted to reiterate our thanks to all the speakers today. I think it's been a really interesting conversation and discussion, and really keen that we can continue this. So I just wanted to make people aware that the project that we've been running for the last 3 years will continue. We have received some funding from Gates Foundation to continue this work. So we're really keen that we can, build on what we've established already, and then define new plans for implementation of this testing going forward. So just to call out to everybody out there, any manufacturers, regulators, etc., who want to be involved as well, please do contact Laura or myself. Our emails are there, or you can go onto the AFSA website and put it in a request there. So yeah, please do stay in contact, and if you have anything to add to the work we're doing, we'd really appreciate talking to you. Okay, Laura, next slide, please. And just before we do close out, I just wanted to mention a couple of events that are taking place. So the first one is in about a month's time, on November the 13th, we're running another webinar on NGS, actually with one of our AFSA partners, PathoQuest. So this is an online webinar, so please go to the AFSA website and you can register there. And then finally, the big event of the year really is our global conference in Bangkok in December the 2nd to the 4th. And this is really to bring everybody together who's been working on this project and you know, create a sort of One Health view really of where we're at with animal testing for replacement of vaccines and where we need to go next really. So we encourage people to register for that as well. Most people we want to be in person, but we will be opening online registrations soon. So yeah, we would really appreciate seeing people at that conference and contributing to that. And that, I think, is probably it from us. And I'll just give a final thank you to everybody who's involved, all the attendees as well. And please watch this space for any future webinars and workshops. Thank you.
Thank you.
Thank you. Goodbye.
Thank you, everybody.
Bye-bye.
Thank you. Bye.
Bye.
Thank you.